So good morning, good afternoon, good evening, everyone from all around the world joining us today. So my name is Amir Mofidi. I am the regional sales lead for Germany and Austria at Biological, and I am very delighted to welcome you to our today's webinar. I see that the people are still joining. In the meantime, before we dive into the presentation, maybe a few quick housekeeping points. This webinar is being recorded, and the recording will be published and shared on our website at cynobiological.com. And if you have any question during the webinar, please feel free to type your question in the chat box, and we will have a Q&A session at the end. To check if our Q&A chat box is working properly, maybe you write or you type the name of the city you're connecting from in the chat box. Oh, Budapest. Someone from Berlin. Houston. Wow, Frankfurt. Wow. Great international webinar today. Yeah, that's great. Yeah, at Cynobiological, we are committed to advancing life sciences by providing high-quality reagents and innovative solutions that empower biological research and drug discovery. Today, we are excited to explore one of the most fascinating innovation in super method that pushes the limits of molecular imaging and allows us to visualize biological structure with remarkable precision. I am very pleased to introduce our guest speaker today, Dr. Kimberly Kramer from Massive Photonic Company. Kimberly earned her PhD at the Max Planck Institute of Biochemistry, where she developed DNA paint workflows to study the nanoscale organization of proteins and RNA. After her PhD, she joined Massive Photonic Company as client solution lead, supporting scientists in using DNA paint kits and solutions to address diverse biological questions. Today, Kimberly will introduce DNA paint microscopy, explain how it works, and share insight into its exciting applications in biological research. So without further delay, I'm delighted to pass the mic to Kimberly. Kimberly, the floor is yours.
Hi, thank you for the very nice introduction. I'm excited to be here and show you how DNA paint microscopy can reveal unparalleled biological insights. So first, I'll tell you a tiny bit about Massive Photonics. So Massive Photonics is a company based in Munich, Germany, and this company was a spin-off from the Ralph Jungmann lab. So Ralph Jungmann is a pioneer in DNA paint development and technology. So he, along with Sebastian Strauss and Jürgen Schmid, founded Massive Photonics in 2019. And essentially what Massive Photonics offers is DNA paint reagents, so plug and play labeling kits or imaging kits for a variety of different targets, different protein targets or RNA, as well as comprehensive research collaboration services. So if you have a project in mind and you want to perform DNA paint, but you're not sure how it works or how to do it, we also offer such services. So now to the talk. First, I want to put DNA paint within the wider super resolution microscopy context. And DNA paint is a single molecule localization microscopy. It's part of that family, the SMLM family. And in this family, super resolution microscopy is achieved by using a subset of blinking fluorophores. So what you would do is you would label your target, here this kind of filamentous target, with fluorophores, and then you need to stochastically activate them. So you don't want to excite all the fluorophores at once because this would lead to a diffraction limited image. But if you excite a subset of these by, for example, different laser lines is one way to do it, you can then image them in a spatially separated manner on the order of around 10 minutes. You take an imaging stack And there are then fitting algorithms that enable you to post-process this data with sub-diffraction limited precision, so you can get the nanoscale locations of these fluorescent emitters in your image. And at the very end of this process, you would then compile all of these image stacks you created over this 10-minute period, for example, into a final super resolution image. And what's very unique about the SMLM microscopy is the blinking and how it is created in each single molecule localization method. In STORM, our direct STORM, you use dyes. So here, let's say your target protein is this blue square, and you label it with this green nanobody that has a dye on it. Here you would do immunofluorescence to label your target with a dye labeled nanobody. And this particular dye would be able to photo switch when activated with a certain laser. And this would create blinking in STORM. Whereas if you look at PALM, which is photoactivatable localization microscopy, you use fluorescent proteins. So here we have our target protein fused with a GFP molecule, our photoactivatable GFP protein, and this protein actually has the ability to switch on and off. And this is how the blinking is created in PALM with proteins and in STORM with dyes. The nature of having the molecule that blinks fixed to your target does create some challenges or photophysical limitations. For example, in PALM and STORM, your fluorescent signal can bleach quite quickly because you're using a dye or this photoactivatable protein. You image it with a laser, create the blinking, but eventually it will go away. Additionally, you're stuck with 2 or 3 target imaging because in here you are actually identifying your targets based on their dyes. So you, for example, you have 1 target in the 641 channel, 1 target in the 561, 1 in the 488. So generally, most microscopes are limited with how many targets they can image in a sample. Target quantification is difficult because you need to fully characterize the blinking behavior of each of the molecules you're using. And this blinking inherently creates difficult quantification, so typically kind of relative numbers are used. Additionally, you can get variable resolutions across targets because you're imaging with different laser lines. So you would, in most techniques, you would have the highest resolution in the 641 area, whereas if you're imaging with multiple targets in STORM and PALM, you might have stronger resolution with 1 target and lower resolution with another, which could create difficulties of analysis, and your blinking can be difficult to control. So with each dye, you kind of need different imaging buffers or blinking buffers, and you have to kind of negotiate these between dyes or fluorescent protein, photoactivatable fluorescent proteins. DNA paint is a method that mitigates or resolves these disadvantages, and that's because the blinking is not created through fixed dyes, it's created through the transient binding of oligonucleotides in solution. So how does this work? You have a target which you want to image. For DNA paint, you need to use immunofluorescence. So let's say you have your target protein and your binder. Here we're using a single domain antibody, which is also called a nanobody. And this nanobody here in green has an oligonucleotide conjugated to it. And this is called the DNA paint docking strand. You would then label this in your sample and add an imaging solution. And this imaging solution would have the complementary sequence to your docking strand, and this sequence would be labeled with a dye. This is now called the imager strand. And when you add this imager or dilute it in your imaging solution, the imager strands are diffusing very quickly, and this is not really captured by the camera, or it's captured as very low background noise. However, when this transient binding occurs, so the imager and docking strand will transiently bind on the order of around 100 milliseconds, 200 milliseconds. This dye is stabilized, so you're able to get a blink that the camera can capture. And this actually happens throughout your imaging acquisition. And because you have an endless pool of imager strands, you have new imagers binding for each event. And this enables you to get really high photon counts and improve resolutions when compared to other methods. So how would this look in raw data? This is a single frame of some DNA paint data. And here you see this characteristic blinking which you would then need to analyze, as I mentioned before, using a post-processing software. And after you do that, you can go from a diffraction limited image, such as this, to a super resolution image. And these are nuclear pores. So when you look at them using DNA paint or other SMLM methods, you should see this 8-fold symmetry, so kind of 8 dots in this circular formation, which we're able to do here. Additionally, DNA paint has several capabilities. One of these is 3D imaging. So for example, here we're looking at microtubules in 3D. So this is the 3D color code. And if we want to see kind of the tubular structure, we can simply cut here on the image and rotate it. So you're really able to see this hollow microtubule structure, which shows you both the resolution and 3D capabilities of DNA paint. Additionally, you can do 3D imaging in kind of deeper samples. So this was in collaboration with Bruker, which has a microscope that enables DNA paint with epifluorescence illumination. So we could do epifluorescence illumination and Z-stacking to look at a whole B cell mitochondria within an entire B cell. And we were able to see this distribution. So this is around 11 micrometers in depth. Another key advantage of DNA paint is its unlimited multiplexing capabilities. And this is, in my opinion, where it can really, really shine. So if you want to image multiple targets in DNA paint, you're not changing the dye. You can actually do unlimited multiplexing with just one DNA paint dye. Instead of that, you need to change the DNA paint sequence. So let's say you have some antibodies on your targets of interest in the cell. Here you have antibody 1 and antibody 2 with sequence I1 and I2. If you would like to image these with DNA paint using exchange paint, which is what this method is called, you would add imager I1 corresponding to the antibody with sequence I1. And this could then bind and create the signal. So this is an intensity over time trace of the blinking. So when you have the blinking, you see here the peak of the signal. So you could then add imager 1, image your target 1 in the sample. And when you're ready to image your next target, if you're using the same dye, you can simply wash out this imaging solution. So how would you do that? You would either do this manually. You can do this using washing buffer, wash your sample. So we typically use EBD8 Will chambers about 3 times, and then you would not see any signal, or you can do microfluidics. So several microscopes have microfluidics options. So here you can always verify that there's no more signal left if you just turn on the camera and see if there's blinking or not, right? Which is very nice. And then you can add your second imager to image your second target, wash that out, and repeat for an unlimited number of cycles. So the imaging has become less dye-oriented and more oligo-oriented in comparison to other SMLM techniques like Storm and Palm. And you can always use more than one dye for multiplexing, but it's just not necessary. And here is an example of a 5-plex image using DNA paint in our multiplexing kit. So in pink, you see mitochondria. In blue, you see endosomes. So here we used LAMP1 as an endosomal marker. And gray, you see the filamentous vimentin structure, so cytoskeletal protein. And you can hopefully appreciate, if your screen is big enough, the spherical structures of caveolin in yellow, and in paxillin as a focal adhesion protein, so it's generally kind of spreading at the cell edges there in red. And you're probably like, wow, that's really cool, but how did you do that if you're doing traditional immunolabeling? So there's actually a trick to multiplexing that you can do that will enable you to use multiple antibody species from the same primary. So what you do, for example, in traditional immunolabeling is you first add a primary antibody to your sample, followed by a secondary nanobody, or an SDAB is what we also call it. And you do this sequentially. So you incubate one, and then in a second step, you incubate the other. There's a new method come out around, I would say, 2022 called pre-incubation. And this is where you can actually pre-incubate your primary antibody and secondary nanobody in a tube together to create a complex. And you do this before adding it to the sample. And there are certain nanobodies that are able to do this. And this really helps you increase the amount of targets you can visualize in your sample. So you would essentially add these in a complex in each tube for around 30 minutes, and then you add them all to the same sample in one amino labeling step. So here we were able to image 5 different targets using primary rabbit antibodies. So hopefully you can appreciate there that they're all separate and there's no crosstalk. Another advantage of DNA paint is that you can actually achieve 1 nanometer spatial resolution within your sample. So of course, another way to think about resolution is it's limited by the label size. So here, essentially, we're achieving label size limited resolution, which at this point in time is really the best you can get in the super resolution field right now. So how is this possible? There was a technique called RESI, introduced recently, and this is RESI is short for resolution enhancement by sequential imaging. And this is essentially a DNA method that you would use if you really want to see individual proteins within clusters or crowded environments. So DNA paint is quite good for up until around 10 nanometer resolution in cells. But if you really want to look at single proteins in these clustered environments, you would need to do RESI to look at oligomerization states, etc. So here on the right, we have a nuclear pore, and in white you see the DNA paint imaging, and in orange you see the RESI. And you can appreciate that in a case where in DNA paint you would just see one circle or one protein, quote unquote. RESI actually reveals it's 2 different proteins because of this higher resolution. And RESI is actually quite easy to implement and quite robust. So if you remember this single molecule localization microscopy trick I mentioned before, where you're imaging just a subset of your target at once, RESI is kind of an extrapolation of that on a single target. So instead of looking at a protein labeled with GFP with just one single DNA paint docking strand, you actually separate the signal into 4 different DNA paint docking strands, so into fourths while you're imaging. And this allows you to then use this SMLM trick kind of twice. And how does that practically look? Is, well, we have a kit, for example, that has a GFP nanobody with 4 different docking strands. And to do RESI, you would do actually normal DNA paint imaging. You would just do 4 rounds of imaging on the same target. And there is an integrated analysis program within the open source program Picasso that I will probably talk about later. And there, essentially, you're able to localize each of these subsets of your target with a subdiffraction precision in your image. And this will enable you to get this super high resolutions. So here's an example of a DNA paint with a nuclear pore. This is the common structure people do these tests on. And with DNA paint, you see this 8-fold symmetry. But for example, when you do RESI, you're able to see that many of these localizations that were kind of one protein or one localization with DNA paint, you can actually separate these into these 2 proteins. And in this particular image, we calculated the resolution, the RESI resolution, to be 0.75 nanometer spatial resolution. So this is really good for clustered proteins, like I said, or if you really need these high resolutions. Another advantage of DNA paint is molecule counting. This is also called quantitative DNA paint. So this is special because you can do not only relative counting, but also absolute counting. In order to do quantitative DNA paint, you need to understand that there are kinetics associated with the binding and unbinding of the imager strand to the docking strand. So here, for example, you see a situation where we have 1 target in our sample or 1 docking strand, and a situation where we have 3 docking strands or 3 targets in our sample. If you're looking at 1 docking strand over a certain amount of time, the imager will bind twice, and this will give us a frequency of 2. And the time when the imager is bound to the target is called the bright time. However, when it's unbound, you get this dark time here. And these parameters can then be used later in the final calculation for QPaint. So here you have a frequency of 2. However, when we have 3 more docking strands, we have now 3 times the frequency of binding, so we have 6 binding events within the same amount of time. So if you look at this paper, there's a really nice breakdown of how they get to this final equation and the calculations. But essentially, using these kinetics, you're able to see that the number of molecules within a certain region over a certain amount of time of your imaging time is related to the mean dark time of your imager strand as well as the imager influx rate. And the influx rate depends on the concentration of the imager strand and the association rate. And these values are readily available in the literature and in your data. So essentially you can just use your data and the literature to get these values. But you need to be aware that if you want to do absolute molecular counting, you need to first kind of create a thumbprint of the kinetics of an area containing a specific number of binding sites. So you get these values with a specific number of binding sites, and then you can extrapolate this to different areas of your field of view. And there are typical calibration structures that people use. One of them is a DNA origami, and this is a 20 nanometer grid origami you see here on the left. These are commercially available. You can put these in your sample, and then you have on this is around, 20 different binding sites, I believe. No, 16. But here you can essentially see individual binding sites and get their kinetics and use this as a calibration structure, or you can look at an isolated target in your sample. A lot of people would use like a monomeric receptor protein to do this. Okay, so I will end this section about kind of DNA paint in general with the advantages of DNA paint-based technologies. You can image an unlimited number of targets with single protein resolution in high and low density environments. Additionally, it's a quantitative method that enables you to count molecule numbers if you would like. The sample prep is a no hassle. We have these labeling kits that fit into your existing immunostaining workflows. And what I didn't mention yet is that you don't need custom microscope hardware to do this. So you can use an SMLM microscope already available in your imaging core, not only to do DNA paint, but also to do the Resi method that enables these high resolutions. So I would like to now switch to a few exciting applications of DNA paint. And the first application I will talk about, because this is a more industry pharma-focused webinar, would be resolving the structural basic of therapeutic antibody function in cancer immunology. And this was recently done using 3D Resi. So Resi, the method that enables these super high resolutions, was used to do this. And I did this because understanding exactly how therapeutic antibodies bind and arrange at the targets or cell surfaces in this case, it's critical for optimizing their function. And the traditional methods that people use really often lack the in situ context or cannot resolve the heterogeneity or really can't identify the targets on the membrane in this case. So in here, they really investigated the structural organization of CD20 receptors under treatment with type 1, example, rituximab. So they use the drug rituximab here, and type 2, obinutuzumab, using anti-CD therapeutic antibodies. And they did this using Resi. So So this is kind of the labeling strategy. And here, really, the DNA paint method advancement was a 2-plex Resi. So we had, in DNA paint world, a 1-target Resi had been done, but here they did a 2-plex Resi on CD20, and they labeled this using an anti-GFP nanobody, you see here, as well as a therapeutic antibody. So they imaged these at the same time. The therapeutic antibody had a tag on it, an alpha tag, and this enabled DNA paint imaging. And essentially, like I mentioned, instead of 4 kind of acquisitions for 1 target, now that they had 2 targets, they needed to do 8 DNA paint acquisitions or rounds of imaging. And this is an example image from this study. So here you see rituximab in cyan and CD20 in magenta. In B, you can actually appreciate the DNA paint versus the Resi results, as well as below in C. and they did this in 3D. So here, it was very clear that rituximab shows kind of a uniform dispersed binding pattern across the cell membrane. And yeah, and you can appreciate that here in the data. When they looked at obinutuzumab with CD20, they saw something different. So here they saw that it lacks this distinct planar higher order structures that they saw with the rituximab treated cells. And here they formed tighter nanoscale clusters of the CD20. So of course, this is great. We can see everything in 3D at these super high resolutions, but we also need to analyze this data somehow. So I'll just give you a quick example of one of the ways they analyze this data, or how they could analyze this data. Yeah, so they looked at the clusters of the monoclonal antibodies along with the drugs using a cluster analysis program. Then here they used DB-scan, which is commonly used, and they were able to compare the ratio of rituximab and obinutuzumab to CD20 within the nanoscale clusters. And they found that there's a 1:2 ratio of rituximab to CD20 in these clusters, whereas there was no linear relationship between the number of obinutuzumab cluster organization and CD20. So yeah, this is one thing they looked at. However, they looked at tons of other stuff that I don't really have time to go in. I just wanted to use this kind of to showcase the type of data you can get when you're looking at clusters and monoclonal antibody mode of action stuff with DNA paint and Resi. So you can look at, for example, the nearest neighbor, the distance distributions between the antibodies and your receptor of interest. You can look at the cluster size and the cluster density, so how many molecules are in the cluster, the stoichiometry. You can do oligomerization state analysis and really look at the 3D spatial organization within the membrane. So you can get a lot of great data, different types of data that could help you with your research. So taken together, these authors took advantage of Resi to directly observe and quantify the in situ molecular changes of CD20 arrangements. And this really allowed them to propose this general model to correlate monoclonal antibody-induced CD20 oligomerization with type 1 or type 2 function. So actually type 2 function, which is characterized by direct cell death, they found that it correlated with limited CD20 oligomerization of dimers, trimers, and tetramers, whereas type 1-like function, which is characterized by less efficient cell killing and these immunotherapies, was accompanied by the ability to form higher order concatenated structures of at least hexamers. And I didn't show it here, but the authors actually also did some functional studies for the type 2 antibodies and looked at different hinge region flexibilities and found that the oligomeric CD20 arrangement determines the type 1 or type 2 function. So this nanoscale characterization really enabled them to understand the structure-function relationships in this particular case. So that's one application, and that focuses more on looking at things at the super nanoscale using Resi. Now I'm going to go into a more multiplexing example. So because DNA Paint offers such high multiplexing capabilities, you can really take advantage of these. And one paper that did so was released last year in Cell by Edward Unterauer, and he looked at spatial proteomics in neurons at single protein resolution. And he did this using a method called SumPaint, which enabled very high throughput DNA paint multiplexing. And with this, he was able to look at 30 different targets. And in combination with an AI-guided analysis, this enabled him to actually discover a new synapse subtype. So how does SumPaint work? Well, for example, in your cell, you have a target or a nanobody. You can label this with a primary DNA barcode here in A. And when you would like to do DNA paint, you need to add a secondary label, which you see here. And the secondary label contains the complementary sequence to the primary label, along with a toehold sequence in orange and the imager sequence in gray. So when this is bound to your primary barcode, you can then do DNA paint imaging. You can then remove this secondary label for various rounds. So in some paint, what they did was they labeled a sample with 30 different antibodies, and they actually did extensive antibody characterization studies in neurons. So this is also a great resource if you're interested in looking at neuronal proteins of immunofluorescence. There's a huge list of antibodies that either worked or didn't work. So maybe that would make your life easier. Yeah, but here they did a primary, they essentially labeled primaries with secondary nanobodies that had this primary label here in A, and added these to the sample and did DNA paint imaging. When they were done with the first round of imaging, they could then remove the signal by strand displacement and blocking. So they used a combination of both. So this toehold mediated displacement is common in kind of DNA nanotechnology, and there are sequences that essentially were able to remove the secondary label from the primary. And additionally, they added a blocking strand, and this reduced crosstalk between imaging rounds. So here, because there are kind of 6 super optimized DNA paint sequences for high speed, if you want high speed imaging, they imaged 6 different DNA paint sequences per round. And at the end of this round, they imaged 6 targets at a time. After imaging the 6 targets, they then removed the signal and went on to barcoding round 2, where they imaged the subsequent 6 targets. And here they did 30 target imaging. And time-wise, this was much faster than conventional DNA paint. Conventional DNA paint, this would have taken around 800 hours, which is a long time, about a month. Whereas with this method, it took around 30 hours. And the student here actually used pre-mixing to enable the study. So like I mentioned earlier, this pre-mixing method. And here is an example of an image. So this is a single field of view, and here are your 30 different targets. And you're able to see in this image a bit more kind of the separate types of targets he looked at. He looked at cytoskeletal proteins along with synaptic proteins. And using this AI analysis tool, they were able to look at the synapses. They looked at over 900 of these and found this new synapse type. So this is really exciting on the multiplexing front, but it's not the only recent multiplexing advancement. So there were additional advancement lately, and I'm just putting this here. So I want to highlight that DNA paint multiplexing is extremely capable, and you can really use this to look at tens of different targets in cells. Currently, the real limitation is actually binders or affinity reagents that work very well. Okay. And I will very briefly now talk about integration of DNA paint into existing workflows before I end this webinar. So what do you need for DNA paint imaging? You need 3 things. You need DNA paint reagents compatible with existing immunostaining protocols. You need a commercial or home-built microscope for a single molecule localization microscopy. These are typically found in imaging facilities. You can also use any microscope that does turf halo epi illumination and has some decently powerful lasers. And additionally, you need a data analysis software, and this can be integrated into your microscope. Oftentimes this is the case with commercial microscopes, or there are really good options that are standalone options. So here at Massive Photonics, we provide DNA paint labeling kits for a variety of different targets. So we have you covered on number 1. So this is just an example of some of our kits. These are, for example, protein tag kits for GFP, RFP, alpha, and EOS. These are secondary binder kits. We have a lot of those. So we are able to provide that. However, we do not sell microscopes, but we have validated many different microscopes with our imaging kits. So this is an example of those. You can actually use DNA paint, and many people do DNA paint mean flux using our kits, as well as other more standard SMLM microscopes like the Abolite, ONI, or Zeiss microscopes, as well as the Bruker-Vutara VXL. And this is actually an Epley illumination microscope that was able to do this B cell imaging I showed you in the beginning. So that's a great option if you're using thicker samples. And the best data analysis tool for DNA paint that we have found and we regularly use is Picasso. And this is actually from the Ralph Jungmann lab. They created it and kind of made it publicly available using a publication in 2017, but you can download it. You can either download the coding version or the desktop icon version if you don't want to do coding. It has a really nice user guide that comes along with it. And most people use 3 modules. So these are 3 of the desktop icons you would see once you download it. And those are localized. So you can localize your single molecule localization microscopy, blinks. You can render them. And then you can also do some filtering by several different parameters. Yeah. So I'd just like to end here and just highlight that DNA paint is a really useful method, and it can really enable you to see a lot and learn a lot from your samples because it has this great multiplexing capabilities, great resolution, and quantitative capabilities. So with that, I would like to end the presentation. And here are the emails and stuff if you would like to contact me, and I'd be really happy to take any questions.
Great, thank you so much, Kimberly, for that fascinating presentation. Actually, it's incredible to see how DNA paint microscopy can deliver such precise nanoscale imaging and, of course, open new doors for molecular biology research. I think now is a time to move on to the Q&A session. Dear audience, please feel free to type your question in the chat box. I think I can start with my question maybe in the meantime. So as a former scientist from the lab, I'm very curious to know how the researcher and the young scientist can start DNA paint imaging, and if there are any typical risk or mistake they can consider before starting that.
Yeah, sure. Yeah, so the easiest way to start DNA paint imaging is to first have an established immunofluorescence protocol. For example, you've done diffraction limited imaging or even storm imaging. So you have a working immunofluorescence protocol. And then use for example, you could use our kit with for the GFP tagged protein. And this really helps you because one of the common mistakes when you're switching to DNA paint in comparison to storm is that when you're setting the focal plane on your microscope, in storm, for example, you have a dye to set the focal plane. Whereas in DNA paint, if you don't have a fluorescent protein, then you need to kind of follow the blinking with your eye. And this is something that takes a little bit to train if you're new to it. So in this way, when you first start, it's very nice to start with a GFP, but if not, don't worry. It doesn't take so long to get trained on this. And if you have, for example, a 3D lens in your microscope, you can use this to set the focal plane very nicely. Yeah, so I would say that the longest part would be kind of getting used to this imaging, but the rest is quite similar. It's for people who have done SMLM before, and if not, it's not too hard, and if you have any issues, you can contact me.
That's great, thank you very much. Jinron asked, is DNA paint is toxic for the cell, and is it possible to use it for live cell imaging?
So DNA paint, we typically use, or you kind of need to use it on fixed cells because it is similar to storm, you're imaging on the time scale of minutes, and you need to do immunofluorescence. So it's not really compatible with live cell imaging. People have been trying to look at cell surface receptors using live cell DNA paint. But in general, we wouldn't recommend this because you are using short, DNA strands. So you're using DNA oligos, which could cause some sort of immune reaction in the cell. So yeah, if you want to combine it with live cell imaging, we would recommend then doing your live cell imaging on the microscope using like a halo tag or a GFP. to track your protein of interest, and then you can like fix the sample on the microscope after you have your live cell data, and then do the super resolution imaging.
That's great. Thank you. And regarding biomolecules imaging, so is it possible to use DNA paint for any other kind of biomolecules than proteins?
Yes. So traditionally, DNA paint has been done on proteins and on structures called DNA origamis, which are kind of used in DNA nanotechnology. So that's a DNA structure. You can also use it for RNA imaging in cells. More kind of the cutting edge thing that people are kind of trying to look into now is looking at DNA imaging using DNA paint. However, this is not something that has a protocol established so far. So that would be an emerging field. Because you can imagine if you're using DNA paint within the nucleus, you're using these shorter sequences, and these might, for example, cause high background within the nucleus. So there are ways to mitigate this as well, such as left-handed DNA or sequence, are looking at the specific sequences very closely.
Great, thank you. Yuchi asked, will the probe target to RNA and genomic DNA and show non-specific binding?
It really depends on how you design the probe. So yeah, I mean, we now have kind of RNA probes compatible with DNA paint imaging. And here we needed, for example, we amplified the signal using DNA paint using a certain way. The most important is that you're able to clearly see your target, and you can also design the imagers in a way where it won't bind to this genomic DNA or RNA, as well as do increased blocking. It's something to always consider when looking at RNA or DNA, especially within the nucleus. But so far with RNA, it has not been an issue for us.
Thank you. And Muriel asked, what are the practical challenges or limitations when imaging a larger number of cellular targets in a single sample, for example, 10 plus?
So I would say the main challenge when imaging large numbers of targets are really screening and finding really well-working antibodies or nanobodies for your targets. Now that we have this pre-incubation, you can use primaries of the same species within the same sample. That helps a lot. And another practicality, of course, is the imaging itself. How are you going to look at all these targets? Do you want to use exchange paint, where you would then need to do 9 rounds of exchange, so 9 rounds of washing in between the images? You can do this manually, or you can do a microfluidic system. So these are kind of the questions you would want to ask yourself.
Fantastic. So another question from Jinran, what's the DNA paint advantages over conventional microscopy techniques for super resolution expansion?
So I would say DNA paint is extremely unique in the multiplexing capabilities over both kind of conventional and super resolution. So you can really image as many targets as you want, as well as the super resolution methods. That's one advantage, along with generally higher resolutions. For expansion, of course, you don't have to expand the sample, which could potentially cause some sort of expansion artifacts. But in general, everything I kind of said in this webinar today, the counting, resolution, multiplexing, it all comes together. Mm-hmm.
That's it. Thank you. Is there any other questions, comments? So another question, how do you resolve for the sub-nanometer scale?
So for sub-nanometer scale, you would need to use RESI, this method. I didn't go over everything today, all the details, but essentially, essentially it's like I said, you would need to separate the target into kind of different kind of subsets of targets, and then look at these with the sub-diffraction precision. And when you merge all of these rounds from the same target into one image, you get this sub-nanometer scale localization precision. Yeah, but the Resi paper is a good resource to look at if you're interested in learning more.
That's great, thank you. So one more question from Mohammed. Do you think if DNA paint might, in near future, move from basic lab research toward clinical application?
I think it's a great contender for this, and I think it will. Recently, there's a lot of advancements in the multiplexing, and people are really looking more into automation for DNA paint and fluidics exchange. So this combined with these advancements will actually enable us to look at a lot of targets within a cell in a shorter amount of time. And this could be very valuable if you're trying to do some sort of spatial proteomics approach like they did in the neurons, or if you really want to see this mode of action with, for example, these monoclonal antibody conjugates. I think it will be really good with that. And yeah, I think it would definitely I think it definitely will move towards this area.
That's great, thank you. And one more question here. How reliable is DNA paint for counting molecules?
So the original QPaint paper actually quantified this, and I believe it was around 95% accurate for both in the cell for counting the molecules. So they relayed this they actually calculated this using a protein with a known number of structures. So they could visually see it, and then they saw it visually also with QPaint. So there they really characterized how accurate it is for counting the molecules.
Okay. And someone asked, how much is this technology limited by fluorescence dye?
Yeah, so here there's not a huge limitation because you really only need one good or great DNA paint dye to do everything I showed you today within the webinar. And that dye is actually Si3b, which is the best DNA paint dye. So people typically use this when they want the highest resolutions. So yeah, you really only need one good dye, so there's really no limitations there. Of course, if you want to make imaging faster, you could use 2 dyes at a time and 2 cameras. So then you could image, for example, Si3b and Atto655 imagers at the same time. And this would help your multiplexing become a lot faster. But yeah, there's not really a limit. It's not limited by the fluorescence dyes.
Thank you very much. I think we have another question here. Is it possible to amplify the signal somehow if the target protein is at very limited concentrations?
Yes, you could do this. I mean, there are different ways to design the docking strand. For example, if you need some sort of signal amplification, instead of having one binding site for the imager, you could have, I don't know, 10, 20, etc. And this is one way you could amplify a signal for a super low copy number target if you don't see it already.
Okay. Thank you very much. Is there any further question? So if not, thank you, Kimberly, for sharing your expertise with us today. And a big thank you to everyone who joined us live and for that fantastic questions. We hope to see you again at our next session. And until then, stay curious and keep exploring. Have a wonderful day and night, everyone. Ciao.